Hello, everyone, and thank you very much for joining us today for this new episode of our Discover Molecular Interactions webinar series. Each month, we will discuss the different molecular interactions and talk about the challenges and also present new innovative assays to analyze them. My name is Vivian, and I am responsible for scientific communication at Dynamic Biosensors. I'm very excited today that I am joined by my colleague Anaï. She's a field application scientist, and she's an expert in nucleic acid binding proteins, but also in the enzymatic activity of nucleic acid modifying enzymes. So welcome, Anaï, and thank you very much for joining us today.
Thank you, Vivian, and hello to everybody.
So I'm very much looking forward to the data that you are going to present today, because we are now going beyond the one-to-one interactions and diving into multi-domain binders. If you have any questions during the talk, please feel free to use the Q&A tool, which you can find in your toolbar. Or if you cannot see that tool, you can, of course, also use the normal chat function, or you can virtually raise your hand and I can unmute you and we can talk directly. So now, without any further ado, I'm handing over to Anaï, and I'm looking forward to your presentation.
Thank you, Vivian, for the kind introduction. And I would like to start my presentation by briefly talk about the technology itself. In this image, you see the Helix biosensor. So it's the machine that we use to measure binding interactions, in particular binding kinetics. The Helix biosensor has all the fluidic, optical, and electronical components needed for an automated measurement. And here you can see the autosampler tray can accommodate well plates, and the biochip rack can accommodate up to 5 chips. And in these chips is where the actual interactions take place. So if we look closer to this biochip, it looks like this. And in the center, it has a single microfluidic channel and 2 gold electrodes, which we also call detection spots. On those gold electrodes, we immobilize DNA nanolevers, these ones labeled with fluorophores, to monitor the binding interactions of a variety of molecules. So these nanolevers are kind of our linker system to immobilize the ligand of interest while the analyte is free in solution. And some examples of interactions that we measure are protein-protein, small molecules with nucleic acids or with proteins. And in this presentation, we are focusing on protein-RNA interactions. Now, regarding the workflow of our measurements, we always start with an empty chip with the so-called anchor strands, which are at all times immobilized on the surface. And then there is an automated injection of the so-called adapter strand with the dye and the ligand strand with the ligand of interest. So in this part, we actually functionalize the surface by DNA-DNA interaction. Then we start recording the fluorescence of the dye, which would be our baseline. And then in the next stage, we start the association phase, in which there is the injection of the analyte molecules in solution. So from this association curve that is coming, this change of signal or fluorescence change is because of changes in the microenvironment of the dye upon the binding of the analyte. So from this curve, we can extract the association rate constant K-on. Then in the following step, we start the dissociation phase, where we replace the analyte solution by buffer. We allow the dissociation to take place, and then we again record the fluorescence change of the dye. From here, we can extract the dissociation rate constant, or K-off, and then we have all the information we need to extract or to calculate the affinity, which is the ratio of K-off over K-on. In a final step, we regenerate the surface of the chip by simply disrupting the DNA base pairing, and so that we can start with a fresh surface for the next cycle of functionalizations. Now, I would like to come back to the actual initial topic of measuring protein-RNA interactions. And we know RNA-binding proteins, or RBPs, play very key roles in the regulation of gene expression. And most RBPs actually comprise multiple RNA binding domains, such as RRMs or RNA recognition motifs, KH domains, cold shock domains, etc., where each of these protein domains recognizes a specific RNA motif. These are also further affected by the cooperation of multiple domains, which enable the fine-tuning of RNA binding and regulation. Now, these binding dynamics are normally not shown in static structures and also equilibrium assays, and therefore it's interesting to actually look at these dynamics with different techniques, and one of them is, of course, binding kinetics. So, monitoring the binding over time. And I brought to you 3 different examples where it's quite interesting to see how these different domains actually interact with each other and give a certain output in terms of binding. All these 3 examples are actually 2-domain proteins. And we always recommend to study this kind of multiple domain proteins to dissect the problem and to study the separate components first. So that's why we did for all these examples. And if we go to the first one, in here we were actually first also interested in looking how this protein binds to different RNA sequences, all of them carrying the RNA binding motifs for the binding of the proteins. So we first look at the first domain with these different sequences, and we see that this domain actually binds to all of them with a very fast association, so very steep association, and also dissociates very fast. So this has a fast on rate and a fast off rate. Now we look at the second domain, and we see already that the association is much slower, but once the protein is bound, it's more stable on the RNA, so it has a slower off rate. When we look at the tandem protein, we see a fairly fast on rate and a more stable off rate, so more stable dissociation. So from this, we can say the individual domains and the tandem protein actually display very different kinetic profiles, which is already very interesting by its own. When we put this data and compare them in the so-called rate scale plot, so this is a representation of the data where the upper scale is the K-on, so the association rate constant. The upper part is the K-off, or the association rate constant, and then the middle part, we have the affinity. So here we have the data for the first domain with a fast on rate and a fast off rate with a KD of approximately 6 nanomolar. On the other side, we have for the domain 2, we have a much slower on rate and also much slower off rate. And when we see the tandem protein, actually we see that both domains complement each other very nicely. And the fast on rate from domain 1 is complemented by the slow off rate of domain 2. So what does this actually mean in terms of binding of the tandem protein? When the protein associates, actually the domain driving the binding is the first domain because of the fast on rate, which leads to a complex. And during the dissociation, we have a rate-limiting dissociation by the slow domain 2. So this is really one domain, the fast domain is complementing the slow domain. Now we can jump to the second example where actually the interactions are quite different. So something very interesting about this second example is that the protein is able to bind multiple RNA motifs along the RNA, and it can also do this so-called looping of the RNA when it binds. So it actually loops around the RNA, around itself. So we started the same. We looked at the separate domains. In this case, we were not able to express the domain completely separate because of stability of the protein. But what we did, we knocked out one or the other domain in the protein. So we see domain 1 actually has very fast on rate and off rate. So from the previous example, we can already infer that probably this domain is the one that drives the binding in the tandem protein. Now, when we look at domain 2, we see a slower on rate and also a much more stable off rate. If you see here the time scale, this is actually in around 40 minutes. So we see that the dissociation is actually really stable. So we can infer again that this domain stabilizes the binding of the tandem protein. Now we wanted to see how these knockout proteins would act in the presence of different or different number of binding motifs on the RNA. So we created an RNA with one binding motif for domain 1, and we vary the number of binding motifs for domain 2 from 1 to 4. And we look at the knockout proteins. So this is the results. Here is what we call a rate scale plot, rate mat plot, sorry. And here the x-axis is the off rate and the y-axis is the on rate. So we see that varying the number of RNA motifs actually doesn't change much for the knockout protein. So all of them are basically scattered around the same points. So we wanted to see now whether this would be also holding for the tandem protein with both domains actually present in the protein. So here we see actually the results for the ligand with only one binding site per domain. And we see already that the kinetics are quite different. And especially these kinetics are what is called a biphasic or a bi-exponential kinetics, which means that there are happening two interactions at the same time, each interaction with its own on rate, off rate, and affinity value. And this can be actually very well appreciated in the dissociation. We have a very fast interaction here that dissociates very fast and then is followed by a slow decay. When we compare these results of the ligand with only one binding site with the results of the ligand with 4 binding sites for the domain 2, then we see a striking difference. So first of all, having both domains of the protein present leads to stronger binding. And second, we can see that the main difference is in the dissociation end level. So actually increasing the number of binding sites for domain 2 decreases the dissociation end level. And this can be explained because the more motifs that we have on the RNA, the more tight is the binding, and also the RNA stabilization because of rebinding effects of domain 2 towards the very closely spaced motifs here on the RNA. Now, the second thing that we wanted to test on this protein was what I mentioned at the beginning was the RNA looping. So this protein is thought to actually loop the RNA around itself when it binds. So we created a setup where we could actually monitor this looping or this RNA restructuring, and we use Foster resonance energy transfer for this type of assay. So what we had was we have the ligand of interest, and then we place a green donor dye and a red acceptor dye on the extremes of the ligand. And whenever we irradiate the green dye, it would transfer its energy to the red dye, therefore we would have FRET. It turns out that this ligand that we use actually was already forming a secondary structure at the beginning, which placed the ends of the ligand very close together, therefore being in a FRET-sensitive distance. So naturally, this ligand actually was in a FRET state. So when we look at the experiments, what we have is that any change in the restructuring of this ligand would actually lead to a change in the FRET signal. So this was quite exciting for us. As I mentioned, here we tested with the ligand with only one binding site per domain. And so the natural state of the ligand was in FRET. So this is the baseline that you see here. Then when the protein binds, it actually opens up the structure when it loops the RNA. Therefore, it separates the dyes and leads to a decrease on the FRET, which we see here in this second stage of the association. So we recover the green signal and we decrease the red signal. And in the dissociation phase, where we replace the analyte by buffer, then the protein leaves the ligand and we have a slowly recovered FRET state. So with this setup, we can actually monitor the kinetics of the RNA restructuring or the RNA looping itself, which is quite interesting to see. So what does this mean in terms of the binding? In the association phase, we actually have a very fast phase driven by the fast on rate of the first domain, followed by the association of the second domain, which leads to a complex. And during the dissociation, we have this intermittent binding of the first domain with these very fast on and off rates. And then we have these rebinding, very strong rebinding effects of the second domain towards the many motifs that we have here, until the second domain dissociates completely. So again, here the second domain is rate limiting in the dissociation. Now I would like to jump to the third example, which is actually also very different from the other two. In this case, we look more at enhancement of the rates and also specificity. So again, we tested first the separate domains. So the domain 1 had very fast on and off rates, here plotted with a KD in the 2-digit micromolar range. And something important to mention for this domain is that this domain is very specific. So we tested the binding of this domain towards its motifs and also towards single-stranded RNA and double-stranded RNA, and it was very specific towards its RNA motif. When we look at domain 2, this already we see that it's very different. So first of all, it's bi-exponential. So we have two rates here, one that is very fast and then following one that is quite slow. And also this domain was completely unspecific. So it was binding to everything double-stranded DNA. So it was not only binding to its RNA motifs. But then when we have the tandem protein, actually the specificity came back. So having separate domains, one of them is unspecific, but having both domains makes the protein specific again. And also, not only that, but the rates were highly enhanced. So the on rates, as you can see, go actually around 2 orders of magnitude higher on rates, while the off rates are mainly the same as the domain 2. So what we can learn from this is that probably the 3D structure of the tandem protein, it's arranged in a way that it only fits probably the single-stranded RNA and not anymore double-stranded DNA or anything, so that it's actually more specific than the separate domains. Another thing that we tried with this protein was looking at how secondary structures would influence the binding. So we tested sequences that were linear with only one binding motif for domain 1 or both binding motifs, and we compared them with ligands that were forming secondary structures. So I'm not going to go into details of all the kinetics, but you see domain 1 is always a very fast binder. Domain 2 has these two phases, and the tandem protein has this also biphasic with a slower component. What we actually learned from these comparisons is that the linear ligands seem to have a better binding than the structural ligands. And this we could explain because in the linear ligands, the binding motifs are more exposed to the protein compared to the secondary structural ligands, where actually the binding motifs were engaged in the forming the secondary structure. So we can see it very nicely in this ligand number 2, where actually the off rate is very, very slow and the binding is very tight in this case. Just to summarize, what we have seen is that multiple domains can interact in different ways. As in the first example, where one fast domain complements a slower domain, they can interact cooperating, so the multiple binding motifs on the RNA actually cooperate to lead to a tighter binding, or they can actually enhance each other and increase specificity. The results of all these interactions are normally having a better affinity and specificity, usually with enhanced on rates and slower off rates. So with this, I would like to finish my presentation, and thank you all for your attention. And if you have any questions, please feel free to ask.
Yes, thank you very much, Anaï, for that inspiring talk about multi-domain RNA binders. And we are now opening the Q&A session. So please send us your questions in the chat or Q&A tool or raise your hand. And I will just start with the first question, which is about the assay setup. The question is whether there's any benefit to use DNA anchors versus using a biotin-labeled ligand and streptavidin beads to attenuate the binding events on the surface.
So this biosensor is based on these DNA nanolevers, and they're actually very convenient to study protein nucleic acid interactions because we can just extend these linkers by the sequence of interest. So it's really a chemical synthesis, and therefore it's really easy to work with it. We don't need to label anything, just order the sequences. So this would be one of the advantages of using these linkers. And also, of course, we use these DNA linkers not only to measure proteins binding to DNA or to RNA, but also binding to other proteins. So this is really the basis of the biosensor.
Yeah, maybe I can also add a bit to that. Like, I think the DNA system also makes it very easy to regenerate the surface compared to other immobilization techniques, right? I mean, you already mentioned that in the beginning of your talk, that it's simply like a regeneration solution and dehybridization process. And I also think, like, especially with streptavidin, because we also do experiments with biotin streptavidin in other setups where we are looking into protein-protein interactions, for example. And it's always a bit tricky because of the multivalency of streptavidin biotin interactions. So that's also, I think, a very good advantage of just having the separate RNA ligand on the surface.
Yeah, that's right. And maybe also a last comment on that. We also have the possibility to use biotin streptavidin in the surface. So if the person has already biotin-related ligands or analytes, we can also put them on the surface. It's not that we cannot do it.
Mm-hmm. Okay, the next question is what the range of affinities is of the RNA binding proteins that we can measure, and whether we have confirmed that with orthogonal methods.
So we have the technical specifications of this biosensor actually cover a very wide range of affinities. We can go from the low picomolar range, 1 to 2 digit picomolar range for very tight binders, to very fast, also very fast binders in the micromolar, high micromolar range. And this is because in the tight binder side, we are able to dissociate for very, very long hours, like 12 hours or so. And in the case of very fast binders, then we are able to exchange liquids very quickly so that there is no artifacts in the measurement.
Mm-hmm. Okay, maybe now that we are already at the limits of the technique, maybe you can mention briefly whether there's a limit for the length of the RNA ligand we could use.
So this depends, first of all, on the chemical synthesis. So around 100 nucleotides, it starts to be complicated to actually produce these sequences. But there is also a possibility some labs produce their own ligands with other techniques that is not chemical synthesis, and then it might be a little bit longer. One just need to keep in mind that the distance between the fluorophore and the binding site of the protein, they should not be too far, because then the sensitivity decreases. So it's one would need to play around, but I would say ligands around 100 nucleotides are still perfectly possible.
How does the switchSENSE system compare with label-free systems? Maybe you can specify what exactly you mean by label-free systems.
So I can comment a little bit on that.
BLI, for example.
So through label-free systems, there is always a discussion in this, right? I would say true label-free is something like ITC, where you really have your things in solution and you don't put any other type of label. We are label-free in the sense that the analyte is not labeled, but we do need to immobilize one of the binding partners on the surface. In the case of RNA or DNA, this might not be a huge problem because you can then just put kind of like a spacer, and then the structure of your DNA or RNA should not change too much. If it is a protein that you want to immobilize, then one would need to keep in mind to not target or to immobilize the protein in a site that is far from the binding site.
Can we eliminate the possibility that the proteins unwind the linker strands?
The proteins unwind the linker strands, or?
I guess maybe it's meant that the proteins cause the adapter strand and the ligand strand to already dehybridize on the nanolever.
I think this is not really a concern, because the hybridization of the linker system, we have like 48 mers, so they are really, really tight hybridized, and unless you are using a protein whose activity is actually opening DNA, for example, a helicase, otherwise it's not really a concern. We have not seen any of these issues.
Mm-hmm. And we would probably just detect it as a decrease in fluorescence, a very sharp, right? If we suddenly lose all our ligand or anchor strand.
Yeah, you would see it in the fluorescence, definitely.
And there was also a second half to that question. That's whether we have problems with proteins sticking to the surface of our chips.
Yes, sometimes, and it also depends a lot on the quality of the proteins. But some DNA or RNA binding proteins, so their nature is to bind these nucleic acids, right? And what we call specificity is always related to the concentration. So if you are way, way above the KD of your interaction, you might see some unspecific binding. But I believe that is actually a very important information for the person to also know that the protein is binding off-target sequences. I think this is also important to know, actually.
Mm-hmm.
I think I forgot to answer one part of a question related to measuring versus some other techniques. So some customers have comparisons between our technique and others like BLI and SPR. Normally we are in accordance with those techniques. Sometimes we are a little bit off, but the trends are always very comparable, especially with the technologies that are very similar to us, like SPR. We normally have very comparable values.
I mean, if you are thinking about a specific system you would like to test, or you have specific analytes where you're not sure if this system could work for you, then please just contact us either via our email or directly via the email address on the slide. And we are always happy to arrange demos and test our system with your specific samples, or we can also arrange some collaborations. So please just contact us and we can discuss it in further detail also afterwards.
Yeah, we're very happy always to test new proteins in new samples.
Mm-hmm. A participant would like to know whether we have a kinetic model information for the two-domain kinetics.
Yes, so we in the software that we use to analyze all these things, that is called Helios, that is also the one that controls the instrument. There we have a variety of fitting models that actually allow the different data to be analyzed. We have monophasic models, we have biphasic models with different free parameters, and the user can choose which one is better for their interaction. I'm not sure if the question was if we have available or like open information, but we can send this information.
Yeah, I mean, the software will also give you all the information, right? Like the mathematical formulas that are used and everything.
Yeah, we're
very
transparent. Yeah.
Exactly. Okay. So if there are no further questions, as I said before, if you have other questions later on after the webinar, then please just shoot us a message. And we're always happy to have a deeper discussion about your specific needs. And other than that, I would just like to point out that there are also previous episodes of our webinars. You can find the recordings of those on our website. You can also find all the upcoming webinar sessions on the website. And if you happen to have some time in September still, then there's also our user meeting coming up. So please also consider just visiting us in Munich for our user meeting. We have a great lineup of speakers this year. So you can also find more information about this on the website. And with this, I thank you all very much for joining us today. And I wish you a very nice rest of your day.